Hello, everyone, and welcome to today's webinar, Simplified Online Tools for CRISPR-Cas9 Gene Editing Design and Confirmation. My name is Louise Baskin, and I'll be the moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. We encourage you to participate by submitting any questions you may have during the presentation. To do this, you just simply type them into the Ask a Question box and click on Send. We'll answer as many questions as we have time for at the end of the presentation. You can also use this to submit any technical issues as well if you have any trouble seeing or hearing the presenter. So I'd now like to welcome our speaker, Dr. Matthew Poling. He's a product manager for genome editing at Thermo Fisher Scientific. And Matt, you can go ahead and begin.
Fantastic. Thank you, Louise. All right, I will go ahead and get started with today's presentation. So for today's agenda, we've got four main topics that we're going to cover. We're going to do a quick review of genome editing and kind of how the mechanisms of action happen at the molecular level, and how we're going to be kind of tying these tools together to allow, users at the bench to have more successful gene editing experiments. The first tool we'll talk about is our TrueDesign Genome Editor, which is our design platform that pulls all of these reagents together into a nice clean package and makes genome editing much simpler for life scientists. We'll then come back and talk a little bit about how we can confirm successful edits using Sanger sequencing and the tools that we've developed to help that workflow. And then we'll kind of wrap up by doing a demonstration of the SeqScreener gene editing confirmation app, as well as kind of some wrap-up and closing comments, and then take the questions from the audience. And to go ahead and get started, we'll do our review of genome editing. And at the most basic level, when we really kind of simplify this down to cartoons, ultimately, what we're trying to do when genome editing occurs is we've got a piece of double-stranded DNA that's represented by those red lines, and some sort of engineered nuclease that's going in and cleaving that DNA in a specific locus. This engineered nuclease can be CRISPR-Cas9. That's kind of become the most popular variant of this. But these fundamentals can also apply to systems like CRISPR-Cas12 or TAL effector nucleases or zinc finger nucleases as well. But ultimately, no matter what enzyme system you're choosing, what we're trying to do here is go in and cause specific double-stranded breaks of this DNA. Now, when DNA is broken in this manner, the cells attempt to repair it, and attempt to repair it rather quickly. Broken DNA is generally not a very favorable physiological condition. And these repair mechanisms aren't always faithful and have opportunities for incorrect repair to occur. And when this happens, we can end up with gene knockouts. By mechanisms of non-homologous end joining that can be imperfect, we can get frame shifts or premature stop codons occurring, which will disrupt open reading frames and disrupt the functional proteins. These will be used to create knockout cell models, or if you're doing something like a CRISPR library screening, this is generally the main genomic readout that you're looking for, and then you'll be screening against any kind of phenotypic changes that might occur. The second pathway for this is going to be my homology-directed repair, or HDR. And this occurs when a donor molecule is provided in excess during the reaction and during the repair process in the cell. These donors generally have left and right homology arms that align around the that will flank that edit site and allow them to be incorporated into the genome at that breakpoint. And scientists will use this to create, kind of reporter genes or fusion proteins. We'll do an example here in this presentation about adding a GFP to the ACTB gene. We can also do this to go in and change specific nucleic acid sequences, like making SNP changes, either correcting SNPs or introducing SNPs to mimic disease models. And this can be a very powerful tool to make very specific changes to the genome. And for either of these examples, you ultimately need three sets of materials to have a successful experiment. The first is going to be that specific and efficient nuclease system. The second is going to be a repair template with that desired change. If you're doing knock-ins, you obviously need to be providing that donor molecule or providing that template at the time of the editing is occurring. And then lastly, it's going to be a method to deliver all of these materials into your cells. We need to be able to kind of package this and deliver this effectively into the nucleus. Now, like we talked about, the most popular version of this that we see, kind of in the life science community is obviously going to be the CRISPR-Cas9 system. Over almost the last 10 years, this has kind of blown up and really advanced our ability to do genome editing successfully because of this RNA-guided DNA nuclease system. If you look at kind of our green bean picture on the right-hand side there, we've got our Cas9 protein and our guide RNA molecules being the two main components. This is adapted from a bacterial immune response pathway, kind of from that adaptive immune response system, where bacteria would try to prevent phage infections and have specific nucleases designed against those various infections. And what we're able to do here with this RNA-guided system is break it down into kind of usable parts, such as our Cas9 protein and the duplex of the CRISPR RNA and the tracer molecules. We use those as single guide RNAs in most experiments. But ultimately, we're able to take these parts, combine them together, and deliver them into cells to have successful editing. When thinking about designing these experiments, we also have to take into consideration some of the context-specific sequences. And for Streptococcus pyogenes Cas9, which is the most common one that we work with at Thermo Fisher Scientific, we're looking at a protostase or adjacent motif or a PAM that's an NGG. And that's going to become important when we talk about how we analyze and look for possible binding sites and possible cutting sites. It's important to keep in mind that the Cas9 system produces blunt-ended DNA breaks. Some of the other systems like Cas12 or TAL effector nucleases will create staggered cuts. But for today's presentation and the tools we're talking about, we're going to be really looking at those blunt-ended breaks and how they're formed, how that can be advantageous, and how we look for that from a sequencing perspective. And then lastly, when we think about how actually one will do this in the lab, what materials they need at the bench, we're looking at using these either as a plasmid or as mRNAs made by in vitro transcription or synthetically, or combining those with a recombinant protein to do an RMP format using a recombinant Cas9 protein and a single synthetic guide RNA. Now, from the Thermo Fisher perspective and how we look and want to be enabling scientists that work with us is we ultimately want to be helping their goal of getting any edit into any cell line. And to that end, we try to break it down into kind of three basic steps for this: design, deliver, detect. And how can we support scientists wanting to move from this design to delivering these into their cells and then detecting if they've had successful outcomes? And for today's presentation, we're really going to focus on kind of the first and the third pillar here. The design piece: how do we design the proper materials to ensure success, have successful knock-ins? And then how do we go about detecting that from a sequencing perspective? We do have a number of presentations also available via lab routes on our website around our delivery platforms, such as the lipofectamine reagents or the neon electroporation system. And we have additional assets available to talk about that if you're interested. Now, from the design perspective, we're going to talk a lot about guide RNAs and donors, but it's also important to talk about the Cas9 nuclease and having the most efficient enzyme possible for that. And to that end, we do have our TrueCas9 enzyme. This is a next-generation protein that has high editing efficiencies. And I'm not going to spend too much time on this, but ultimately, you want to be starting experiments off with something that's going to have very high efficiencies and be able to be effectively delivered into your cell types. Using something like a plasmid can be effective for some examples, but ultimately we've shown, in numerous examples that delivering a Cas9 protein can lead to higher editing efficiencies and overall higher success and experimental outcome. And that's why we generally recommend a Cas9 protein for all of our protocols and approaches here, and you'll see that as we go through the tool as well. Now, to think about designing experiments here and designing those sequences, it can be misleading to think that you can just use any guide RNA for any CRISPR experiment and get success, because not every guide RNA is actually appropriate, and not all guide RNAs are kind of created equally. The stem, basically the basics of a guide RNA will be the same from experiment to experiment, but that seed and spacer region can be different for each experiment, and those can significantly affect the outcome. And to kind of break it down, we're looking at two main variables here. One is the efficiency. How well is this guide RNA going to target and cleave that locus? How well and how can we predict these outcomes in silico before even needing to go to the bench? How close, what's the GC composition of that region? What kind of repeats are there? Is there possibility of any secondary structure? Because when we use inefficient guides, it can lead to, kind of wasted resources and time and ultimately frustration because of a lack of success. So you want to be going in when using something where you know you're going to have that good efficiency and high likelihood of success. The second piece of this is off-targets. So while we assume that guide RNAs are going to be specific for the locus that we've designed them to, we and others have often found that off-target effects can occur. Even if you don't have perfect base pairing, some single base substitutions can still even lead to editing at unintended loci. And when we don't take this under consideration or control for it, you can actually disrupt other major genes and significantly affect your outcomes and confound results. It can be very difficult to make meaningful interpretations from the data. And if you're an investigator thinking about going into clinical applications, this can be even more important as these off-targets could be genotoxic and ultimately, damage the cells that you're editing, or oncogenic and creating unintended, cancerous phenotypes. And so off-targets can be a very important consideration as well, and we can address that with in silico predictions. And so ultimately, how does one contend with this? It's really using, kind of the knowledge from these data sets to build an in silico prediction model. And at the basic level, how this will work is that we'll look at a nucleic acid sequence here. We've got about 51 bases on the screen here, of our double-stranded DNA. And the first thing we'll do is we'll look for those PAM sites. We'll try to identify that NGG sequence on the top and the bottom strand that are going to be suitable for Streptococcus pyogenes Cas9 and guide RNA binding. We'll take those sequences and we can do alignments on them and we can start to rank them. And then we can ultimately do binding and off-target predictions using our software here. So we can go through and make predictions of, these guide RNAs are going to have really high scores. We think that they're going to be highly efficient. And then look at those number of off-target effects. And we can do kind of this forced ranking and weighted prioritization of binding and possible off-targets. And then ultimately provide recommendations to the user as to which ones we think would be suitable given their experimental inputs. Now, the other molecule that's going to be important for this as well, especially if you're going to be doing those knock-in experiments, is the donor design. And for that, we've also, been hit with a lot of questions on that end as well of, like how close does, that edit site need to be to that cut site? How long do the homology arms need to be? Should we be using something that's symmetrical or asymmetric? Should we be using single-stranded or double-stranded? And ultimately, we kind of, end up in the same general question of, can you help me design my donor? I don't know what I need to be doing here. And to that end, we, really the origin of the software that we built here was really to help address this problem. And we acknowledged that we needed to establish kind of like consistent and simple rules for this, make this design tool easy to use. We want to be helping, investigators, who are just beginning their journey as well as our experts. So we need some sort of intuitive interface that makes it simple for everyone. And we want to make sure that this is free and available to scientists globally. And being from Thermo Fisher, we have the opportunity to do that with our cloud platform. And so we made this free software available on Thermo Fisher Connect, and I'll be demonstrating that. And then you also might be thinking too, well, you've come up with these rules, but how do we know that they work? And to that end, we've actually published in a peer-reviewed journal our design considerations for this. How do we need to be designing donor molecules to ensure success and make it as simple as possible for users? And so we published just a few years ago in the Journal of Biotechnology, and this article is available, or is linked, on our marketing page that I'll be sharing, and it is available for anyone who wishes to go view it. So that information is available as part of the resources. So to that end, and some of the data from this publication, this is not an exhaustive review of that, but looked at trying to understand the proximity of these cut sites using a single-stranded donor. So across the top here, we've got about a 70 base pair region with position 0 being marked as our possible insertion. And we're doing a 6 base pair insertion to repair GFP. So ultimately, when we get successful insertion here, our cells will turn green, and that is our readout. And so we see that we have possible guide RNAs from the -34 position all the way up to the +30. And when we look at our cleavage events across this entire span, we can see that we get a variety of guide RNA effectiveness. If we look at our percent Zindel, we're averaging around 50% overall, with some getting closer to 80, and others like our +21 guy being, pretty poor, less than 20% editing. But overall, kind of across this range, we've got, kind of average outcomes here from gene editing, from an indel perspective. But when we look at our ability to repair GFP to have the successful 6-base pair insertion that's going to allow that protein to be functional, we really only see success when we're within 10 bases of that insertion site. So remember, the insertion is at position 0, and if we look from -7 to +8, that's where we start to see significant differences here. And we're seeing that GFP repair and that knock-in rate actually start to go up several-fold. And so if we kind of think about this holistically, when we're flanking within 10 base pairs of either side, that's where we really start to see this effect and improve. And even, and you can also see that there's a secondary issue here with that +3 guide RNA that doesn't perform very well. We actually see that knock-in rate drop significantly. And so kind of with these data and supplementary data as well that I'm not showing here, we kind of try to come up with some guidelines and general rules, and the first being ensuring that your insertion site is within 10 bases of that DNA break, and utilize kind of the highest cleaving Cas9 guide RNA pairing that's available for your design, and trying to kind of ensure your success by having those tools be complementary and high efficiency. So to that end, we kind of have our 3 simple rules for genome editing. You'll kind of see a trend here where we try to break things down into 3 things: design, deliver, detect, our 3 simple rules. But ultimately, we've got our first rule being have your DNA break less than 10 bases of your desired edit site. Ultimately, wherever you want to be causing that edit to occur, you really want to be using a design that's within 10 bases of either side of that edit site, because we really start to see the performance drop off when we go outside that range. And so we have rules in place to try to keep you localized around that locus to have that efficiency be the highest. The second is to use the most efficient enzyme system possible, so that'd be using the highest scoring guide RNA available. It might be changing out your tool set and considering something like TAL effector nucleases, if that'll be predicted to be higher editing than the CRISPR-Cas9 system. And then the last is use at minimum of 30 nucleotide homology arms. We really want to keep the system compact and easy to use, and we found that 30 nucleotide homology arms are sufficient to get high knock-in editing, and that really around designing, as close to that edit site as possible is really going to be delivering the higher level of success. And so using minimal homology arms when using a donor can be a very effective tool. So to that end, what we've tried to do here today, what we tried to do is build our TrueDesign Genome Editor platform. And this is a software that lives on Thermo Fisher Connect. I'll be doing a demonstration of that in just a moment. But what we ultimately want, or what we're supporting our users with, is searching for their gene of interest. We've got 5 different species in the software now: human, mouse, rat, zebrafish, and roundworm. Allowing them to go in and find the specific transcript that they're looking for, find the amino acid and nucleic acid sequences they want to modify, and make those specific changes, and then ultimately giving them a complete design, giving them the Cas9 guide RNA sequence, giving them the donor molecules, so that they can have a complete solution and have a successful editing experiment at the bench once they receive those materials. So for our first example, I want to talk a little bit about how we support this with fluorescent tagging. And what we have here is our TrueTag Donor DNA system. So this is our solution for doing kind of larger insertions such as tagging. And the way that the system works is that we have N-terminal or C-terminal templates that can be customized to the locus of interest by PCR. And so these templates are provided to the user, and by a simple PCR reaction, we can add those homology arms, which you can kind of see in orange there on our schematic, to these donors and make them specific for that particular locus and a guide RNA. And to this end, we're able to get editing efficiencies, or excuse me, get high editing efficiencies and get up to 99% edited cells using those resistance markers that are built into the donors. By applying puromycin or blastocidin selection, we can really drive that cell population to being greater than 90%, almost 99% edited cells. And you can see an example here in the upper right-hand side of some photographs of GFP-tagged GFP-tagged cells at the actin locus, and nice actin filaments actually being stained green from that GFP, an endogenous GFP in that system. And then looking at the graph from the lower right-hand side, you can see how we were able to drive that editing efficiency and that overall edited population to being greater than 80%. Now, the way that this is going to work from kind of a textbook illustration perspective is that we have our single guide RNA, we have our Cas9 protein, we have our TrueTag donor that we've customized by PCR to our locus of interest, all being combined together with the CRISPR-Max lipofectamine reagent. And once we create these little lipid delivery vessels, we'll be transfecting them and delivering them into the cell. These materials will be delivered into the cytoplasm. They'll be taken up by nuclear import and ultimately go in and modify the genomic locus. So we've got our Cas9 going in and cutting the DNA. We've got our donor molecule being incorporated at the locus of interest, and then ultimately leading to the expression of a GFP tag protein, as you can see there in step 7. And so what we're trying to do is, again, trying to simplify this process and really provide all of the design considerations and all of the necessary materials for this to be successful upfront in a single tool. And then again, to kind of briefly go over kind of the illustrations here of how we use TrueTag, we start off with kind of a basic donor where we then use left or right homology arm PCR primers to extend those out and make them specific to the locus of interest. And this is an example of doing N-terminal tagging, so doing it to kind of the front end of the gene at the start codon, or for C-terminal tagging and doing it at the back end of the gene near the stop codon as well. And these illustrations are available within the TrueDesign tool if you're looking to see more of them. And without any further ado, I'm going to go ahead and switch over to my browser for a demonstration of how to get into the tool and how to use the software. So give me 1 second while I change my screens. Okay, hopefully I am now sharing my browser for all of you, and we can go ahead and look at where this tool lives on thermofisher.com. So this page can be reached via thermofisher.com/TrueDesign, and this is our landing page for the TrueDesign software. When you first come to this page, you'll be greeted with a button that's in bright red here that is to launch the application. And then we also have a number of assets available on this page as you scroll down that speak to the capabilities of the software, some screenshots of the workflow, as well as additional resources for getting started. Now, for the sake of time, I'm going to go ahead and go straight into the tool by clicking the Launch the App button. This will go ahead and open a new window for you, and then it will also then redirect you to our Thermo Fisher Connect login page. Now, if you have an account with thermofisher.com already, you can go ahead and use that username and password to sign in. If you don't have a login with us as of yet, you can go ahead and create an account with just a few steps, and I'll demonstrate that here when we click Create an Account. So when we go ahead and do that, we are going to be just asking for your first name, last name, and email address to create an account. You don't need your full billing and shipping information, really just the basic logins that you need to access a cloud tool. So for this example, I will just use a Gmail address here. Oops, if I can type. And then we'll go ahead and give a strong password that no one can guess. And then the last thing we ask for you is to say if you're interested in receiving more additional information from us about our products and services. Obviously, we like to be able to reach out to all our scientists that we support, but if you're not interested in receiving email communications from us, go ahead and feel free to click that No button and create an account. We'll just take a moment to process. All right. Alrighty here. Looks like our registration was successful. And what we can do at this point is you can go ahead and complete the rest of your institution info, but you don't need to do that at this stage. We can go ahead and go back to our TrueDesign Genome Editor tab, click Launch the app, and then we'll go ahead and be presented with the tool. Once this page loads, oops, just give me 1 second here. All right. And as we're accessing our Thermo Fisher Connect platform for the first time with this account that I just made, we're going to be accepting our terms and conditions of use for the platform, as well as specifically for the TrueDesign software. All right, now we are in TrueDesign and we are ready to do our first experiment. So as you can see, we're presented with 5 different experiment options: gene knockout, fluorescent tagging, insertion, deletion, and SNP edit. For our insertions and deletions, we are limited to 30 bases. We are supporting designs of that smaller side because we're trying to keep everything contained within a single DNA oligo. And to that end, we can support up to 30 bases plus those homology arms that I was speaking to. For this first example, we'll do fluorescent tagging. So I'll go ahead and select the button and click Next. We'll then have our 5 species to choose from. We've got human, mouse, rat, zebrafish, and C. elegans, the roundworm. I'm going to go ahead and do the human example. We can then search by gene symbol, DNA sequence, or chromosomal locus. I'm going to do gene symbol. And then what the tool will allow you to do is it has some predictive text recommendations. So as I start to type in ACT-B to go for the actin B gene, you'll see that those lists start to refresh. And we can see that the first one on our list here is that ACT-B gene. We go ahead and search for that, and it'll pull all of the relevant transcripts for the actin B gene on NCBI. And so we can see we've got our transcript here on the list. We've got our, if we click on that hyperlink, it'll actually go ahead and take you out to the NCBI sequence for your gene so that you can cross-reference and confirm that you're ultimately going to be working with the right gene here. We know this is the right one, and so we're going to go ahead and select Edit. So now that we're on our second step of the software, we're on our Edit page, we now have a number of new features available. Starting at the top of our screen here, we have a topology map of the ACT-B locus. We can see our introns kind of labeled here. We've got a very large exon 4. We've got our exon 6 here. And if you click these plus minus buttons, you can kind of zoom in and get better definition of this topology. And so we can see here we've got kind of our exon 2. We've got a little bit of an untranslated region, our intron, and the following exon. And if you go below that, the sequence editor is going to be showing a detailed view of what's in that blue box. And so we can see, again, here are annotations for intron 1. We can see our second exon here, and we've got a little bit of untranslated region before this methionine of our start codon. So for our tagging experiment, we're going to be doing choosing from these pre-selected options. So the first thing that we're going to choose is our doing an N-terminal tag. So we're going to choose amino N-terminus. We could also choose carboxy terminus if that's the position you want to be tagging. We're going to choose between our GFP or RFP reporter, and then we're going to choose our blastocidin or puromycin resistance marker. Once we have our selections chosen, the Add Tag button will turn red, and it'll go ahead and insert that into our genome, basically at that start codon position. So we can see our Puro GFP arrow here right after that start codon, and we can see that summarized here on our edit list. If we want to go back and make changes, we can click this undo button, and that selection will be taken out, and we could switch this over to RFP or switch to the blastocidin marker. But we'll keep with those initial selection choices, and we add them in. And then we'll go ahead and click Design. And this is where all the bioinformatic fun part comes from on the back end here. So what the software is doing at this point is it's scanning this region, looking for possible PAM sites near that edit site. If you recall, we're trying to keep everything within 10 bases of our prospective edit site, which is going to be right after that start codon. Once we've identified all of those guide RNA sequences, we're going to start to do blast alignments for them, try to predict where those off targets might be, what the prevalence is of those, what the scores of those off targets might be, to give an overall ranking and recommendations of guide RNA targets. Once that's done, the last thing it's going to do is it's going to go ahead and design those forward and reverse primers for those large knock-in donors that will do that GFP knock-in for us. And so The software will kind of run through and do all of this bioinformatic power on the back end using the Thermo Fisher Connect Cloud, and then ultimately present the results to us. And as you can see here, we now have our 2 possible guide RNAs that were identified being within 10 bases of this insertion site. We can see our first one here has our green check mark saying it's recommended. If I click on this row, we can see it showing up in our sequence editor here, the green box being kind of that binding sequence, and the purple box being our PAM site. We can see our score here of 95.9, so a really nice high-scoring guide RNA. We've got 9 possible off-targets. If I click this hyperlink, it'll show a pop-up modal that has all of those sequences and possible off-target sites here. If you see one where it does have a 0, also in chromosome 7, this is actually our on-target site here, and basically showing that we are successfully identifying our on-target locus. And then if we look at the far right side of this table, we're going to have our fluorescent tagging primers with that forward and reverse primers that'll customize that donor template to be for this actin B locus. You can see we've got a second guide RNA that came up here. It does not have our green check mark, and that's because we have a score of 35. Even though it's a little bit closer to our edit site, the GC composition here is considered unfavorable for editing efficiencies, and ultimately we get a score of 35, and this is really not the best choice to move forward with. And we can see this guide RNA as well, see that it's on the bottom strand by selecting the row, and it's highlighted up here in the sequence editor. We're going to go ahead and select this guy by checking our blue check mark here, and then we're going to go to our summary page. Now on our summary page, we're going to have all of the design inputs that were put in summarized for you, as well as some product selections to ultimately pull this all together. Starting from our top left-hand side, we have our our Cas9 format. We have our TrueCut Cas9 protein or our CRISPR nuclease mRNA. We have our single guide RNAs, our synthetic guides in a 1.5 nanomole scale. And then we also have our transfection reagent, that CRISPR-Max reagent I was speaking to earlier, that's going to be used to bundle this all together and deliver this payload into the cells. You can view your CRISPR designs in donors in this little pop-up modal. So we'll see we've got our ACT-B CRISPR number 1, we've got our sequence here, and then we've got that forward and reverse primers being presented as well. We also have kind of our total product summary here of everything that's needed for this to be successful. And then we also have additional products such as sequencing primers, which will come into play when we talk about Seq Screener in the second half of this presentation, as well as positive and negative controls, which are essential for properly controlling for the experiment. If you wish to view the illustrations that I showed earlier in the presentation in PowerPoint, these are also available within the software and can be used for review or for presentations, lab presentations, or being saved for notebooks. And then lastly is to export your results and protocol. And if you click this button, you'll go ahead and have a download that shows up and is saved to your local disk. That'll have all of those gene editing outcomes saved in a downloaded Excel file for future reference. Just wait for my Excel to open up here. Fantastic. So you can see we've got our guide RNA sequence here. We've got our forward and reverse primers. If we go to our gene editing design information, you can see all of the input information, kind of our specific locus, the transcript we're working with, that gene symbol. And then kind of the last thing to call out here is that we also have a protocol helper. So we have an overview of the CRISPR-Max transfection protocol, as well as a complete transfection table. And then lastly, if you're ready to move forward and acquire these materials, you can click Add to Cart, and all of the materials will be added to your thermofisher.com cart. So let's go back and do another example here real quick. So instead of doing fluorescent tagging, I'm going to go ahead and do a SNP change. And for this example, we're going to do something in mouse. Now, to search for this gene, I'm going to actually use our DNA sequence search option. And what this allows you to do is to enter a 500 to 200 base pair region. I'm going to copy this from my other window that's off screen. I've got I believe this is about 120 nucleic acid sequence, and we're going to go ahead and map that sequence against the mouse genome, and it's pulling up the myostatin mouse gene. And so we see that that's our transcript that we're looking for, and we're going to go ahead and edit that. Now, what's happened here is that since we put in the sequence that corresponds to exon 1, we're actually being delivered and dropped into that region of the mouse gene. So we're already centered on this part of exon 1. Now, what I'm going to be doing here is I'm going to be selecting this particular base at position 2008-6. Oops, let's see here. I meant selected, didn't quite go. There we go. And we're going to be doing a specific G to T mutation here that's actually going to be inserting and converting this to a stop codon. So we've changed that into a stop codon position here that will ultimately disrupt the open reading frame of this myostatin gene in the mouse. And so if we're doing this like in mouse embryos, we could be making, kind of those mice that have excessive muscle formation. It can be kind of an interesting model for gene editing. But we've got a single stop here, and we maybe want to be ensuring that we're really going to be truncating this protein and have absolutely no risk of any possible read-through. So we might do a second edit on top of this, and we'll do a secondary insertion right after this stop codon we just put in, and we're going to put in 2 additional stop codons. So I'm going to do TGA and TGA. I'm going to go ahead and insert that, and now we've got a triplet of stop codons that have been inserted at this secondary position. So again, we did a SNP change at this first position, and then we've done a secondary 6-base pair insertion at this secondary position, just 3 bases down. So this is a rather complex edit that we're supporting here with the tool, and we'll go ahead and see what comes out the other end in terms of guide RNA design and what our donors look like. So again, we'll go ahead and click the design button, and the software will go through and do this entire bioinformatic analysis for us, looking at possible guide RNA sequences, possible off targets for that, as well as those donor molecules. And with that, we'll let this run, and I will take a sip of water and catch my breath. All right, and there are our results for this particular design. So we've got our 2 edits being incorporated into our donor molecule. We have our single guide RNA that's being recommended here. It does meet our thresholds of being suitable and getting that green check mark. We've got an 85.8 score here. We do have a higher number of off targets, but since we're trying to do some very specific changes here, this is going to be our best recommended guide for this knock-in experience, or for this knock-in experiment, excuse me. Again, we can see the guide RNA binding site relative to these insertion sites, and we can go ahead and select that to move forward with. A little bit different for this view since we're doing a SNP change. When we go ahead and view our donors, we'll see our guide RNA sequence here, and then we'll also see that donor molecule with those specific changes being presented as well. And then we can also do the same thing as well with this, where you can view your gene editing experiment. Our illustration is revised to reflect these kind of SNP insertion deletion changes, as well as export these results to save them to your local disk, or you can add all of these products to your thermofisher.com cart if you're ready to try them in the lab yourself. With those 2 demonstrations, I'm going to go ahead and switch back to my PowerPoint. Just give me 1 second while we load that back up. All right, now that we're back in the PowerPoint, we'll review briefly kind of the benefits for the TrueDesign Genome Editor. This allows users to search for their gene of interest. You can go ahead and craft the specific changes you're looking to do, and then ultimately kind of complete your design and have a complete package of materials to have a successful editing experiment at the bench. Again, this can be found at thermofisher.com/TrueDesign. Now, once you've done this experiment, you need to think about how you're going to go about confirming it. How are we going to actually determine if these results were successful? And so we've delivered Cas9 and guide RNA to our cells. We've made a pool of mutant clones, but ultimately, what's happening here? We've caused that double-stranded break, and we've had DNA repair occur, but did we get the outcome that we needed? And ultimately what happens here is you'll end up with a pool of edited cells. You might be working in like a 24-well dish, and within that single well of the dish, there's going to be a pool of various editing outcomes that occurred. You're going to have some cells that might be wild type. You're going to have cells that are going to be homozygous, that have both alleles have the correct edit. You might have cells that are only heterozygous, that have one allele corrected but one allele different. And how are you going to kind of tease those apart? How are you going to understand the editing efficiency? And so there's a need to do kind of a pooled screening or do kind of a pooled analysis to verify what kind of editing efficiency and what kind of outcomes you have from that pool of mixed population of cells. And then after that, you'll need to go into a clonal expansion step where you're going to be kind of pulling out either your heterozygous, your homozygous, or your kind of isogenic wild-type controls for further analysis. And so for this clonal screening, there's also a need to understand kind of these various allelic states. Do we have the cells that are wild-type in this population, this particular wild? Do we have ones that have both alleles edited or just one allele edited? Was our clonal isolation protocol not super clean and we have a contamination where we have a mixed population of cells here, even though we were trying to do single clones? And the way that we can go about doing that is actually through Sanger sequencing. So Sanger sequencing is actually a pretty straightforward process at this point, something that we've really optimized at Thermo Fisher Scientific. And what you're going to be doing is doing a simple PCR reaction and a cleanup, and then going through a cycle sequencing reaction that's going to ultimately create the various lengths of DNA for a Sanger sequencing run. That material gets further cleaned up, and then it's going to get run on a genetic analyzer through capillary electrophoresis. And through that process, we're able to do specific base calls to understand the nucleic acid sequence as it gets run through that capillary. But kind of the new piece to this is doing that data analysis, because it's actually not quite as simple as I just said. So when you have your edited sample and it kind of comes out the other end of the capillary electrophoresis system, if you look kind of to the right of that dashed line there where our double-stranded break occurs, you can start to see that the base calls become kind of jarbled. We kind of mix peaks where we've got, maybe it's clearly an A and some are maybe clearly a T, but other ones you can kind of see multiple base calls coming through. And ultimately, as you read across the top, we start to see more and more degenerate base code showing up as the reads here. And this is because ultimately when the DNA repair occurs, we don't have kind of this unique clean sequence to read anymore after that break site, after that edit site. And so how are we going to align and interpret these results? And how can we do this from a software perspective? Well, the deconvolution of all of these reads has been done by a number of different groups, but we've been able to take that base software and expanded it to create it to make a more useful piece of software for the genome editing scientist base. And so what most tools have is kind of an ability to do some primary screening and kind of do batch mode analysis where they can take a lot of samples, but they don't really have the ability to support that secondary screening workflow when you're thinking about looking at clones and how do you do that. These softwares can be pretty slow. They take a long time to analyze the data, and they don't really save the information on the local disk. It's generally you load it to a website, the information's there, you need to capture a screenshot because it's not going to be there when you come back later. And so they're not connected to any kind of data repository or ability to save this. And so what we wanted to do with the Seq Screener platform is kind of create something that's going to add additional value and additional be more user-friendly for the genome editing users. And one of the most common things that we will get asked from users is, kind of how does this compare to next-generation sequencing? Are we seeing equivalent results? Can we trust these results relative to next-generation sequencing, which, has been kind of considered, the standard for a lot of these outcomes, but is a much more labor-intensive and complicated process. And so we did some benchmarking here, and you can see that we've got some really nice linearity in terms of editing efficiencies for Sanger sequencing across the x-axis versus NGS across the y-axis there, giving us that we have some pretty good confidence in the editing outcomes that this Sanger sequencing tool can give us. And to that end, we built out the Seq Screener gene editing confirmation app, and trying to use kind of the same look and feel and intuitiveness that we have with our Thermo Fisher Connect platform, building a software that allows for quick identification of successful edits We want something that's going to be fast and accurate. We wanted our algorithm to be robust, to have that strong linearity with NGS data, which we use as our baseline, like I showed on the previous slide. And then having it kind of widely available by having it on Thermo Fisher Connect. Having this be a platform that any investigator can access with just a simple login like I demonstrated before. And so again, really wanting to have, multiple pieces of software and having multiple solutions to support, gene editing users here. And with that, I'm going to go ahead and switch back to my browser, and we'll do a demonstration of the Seq Screener software. All right, hopefully I am sharing my browser again, and now we have our Seq Screener gene editing confirmation app up. As I'm still using that new account that I just made for the Thermo Fisher Connect platform, I'm going to be accepting these terms of use in the EULA, and we'll be saving this information onto the Thermo Fisher Connect platform. I'm going to go ahead and close this out, and we'll close the quick tips because I'm going to show that to you. And kind of the way that this software is set up is you're going to be able to save your data into the platform, and we're going to be importing edited samples as well as control file samples here to be doing these comparisons and these analysis. So the first thing I'm going to go ahead and do is I'm going to go ahead and pull up my data sets here. So for our import our edited samples, I'm going to be dragging in a folder of samples here, and this is going to have 6 samples within it. And I'm going to go ahead and control the matching control files for all of those samples. And the software is going to go ahead and take those AB1 files coming from the genetic analyzer and validating them, and then presenting these out, basically these edited sample files, in a table form here. Now we do need to map these out so that they know which control file they're going to match with or which guide RNA was used for cutting this. We also have the option for aligning and analyzing a donor sequence as well. So you're able to put in your guide RNA sequence and a donor sequence, and the tool will take those under consideration for predicting the editing efficiency and doing that analysis. The fastest way to do this for a lot of samples is to go ahead and upload a mapping file, and I'm going to go ahead and just do that from my saved files here. I'm going to go back to our primary sample here. There we go. And we're going to go ahead and load those files. And so you can see that using our mapping file here, we've got all of our various guide RNAs that we're testing here loaded up. We've got our matched control files, and then we've got our sample files here. And we can go ahead and hit Analyze. And now the software is going to go through, and you'll see how fast it is to do all of these analyses and provide us with meaningful outcomes. So when you end up on your results page, you'll be able to see kind of at a quick view here, kind of the grading of the wells. So basically how we're scoring these, what plate it came from, what well on what plate it came from, how we've titled it, whether we've analyzed this as a knockout or a knock-in, those target sequences, and then kind of the main outcomes here are model fit and our editing percentages and kind of our frame shift editing. And then the last thing here being diversity of edits, or the number of edits that have been identified in this sample. Starting off with our grade, if we hover over this, you'll see that we have recommendations for, kind of like ideal, good, or needs review. Ideal meaning samples that are going to have, very high confidence, high model fit scores, high editing efficiencies. We have very high confidence that the sample that you're analyzing here was a good knockout and is something that you could be considering and propagating further. Good knockout is kind of in that middle ground with something where we have that high editing efficiency. Our model fits good, but it's not as high as kind of that ideal knockout model here. The remaining ones are going to be kind of in the needs review. Something like a model fit might not be great. Like if you look at this one here, the sample 4, our model fit score is 0.44, so we don't have a whole lot of confidence that these reads were clean enough to make that call. And then the ones marked in W are going to be wild type, and we can see overall we have either very little to almost none editing efficiency. Less than 5% here. This is basically a wild type sample, and 0.77 here. So we had very poor efficiencies of this guide RNA. If you want to go in and look at any of these examples, you can just click on the row, and it'll take you into an overview of the plate map, and this will be important when we do the clonal screening view. But really what I want to show you is this indel frequency chart. And overall, what you can see here is that we are displaying out bar charts of the predicted frequency of that editing outcome in this sample. So you can see here that we've got, kind of a +1 insertion contributing almost 65% of the data that came out of the sample. We've got a -1 being about 15%, and we've got some minor variants as well that are also showing up. If we scroll down, we can kind of see the alignment here of our control. Here's our break site in the dotted line, and then where this +1 insertion here. So we've got that plus sign here, and then here's how the rest of the sequence is aligning. And then looking overall here for basically anything over 5%, we have it highlighted here, kind of our +1 being the major contributor, -1 being a secondary contributor, and kind of this -10 also being a tertiary contributor to this editing efficiency. Going across the top here, we see our model fit is 98. So this is a pretty strong linearity. We have high confidence in this data. Our overall editing efficiency is going to be 96.25. So that's the sum of all of these variants basically that are not 0. And then our percent frame shift, which is accounting for everything that's not going to be a +3 or a -3. So, a +3 insertion might have the same reading frame as the wild-type protein, just with an additional amino acid, or a -3 might just have removed a single amino acid. And so we're considering here, how this might be overall disrupting the reading frame and the production of a functional protein. And then the last thing you can do here, too, is you can look at the sequencing traces. Oh, there we go. Rid of the quick tips here since you have me demoing it for you. And we can see the actual traces that came out of the genetic analyzer and being able to see kind of how this data, kind of became convoluted and kind of messy after our edit site relative to the wild type control, our unedited sample where we have really nice clean reads throughout the sample. And so you can see here kind of how messy this data is and how our software is able to kind of pull out meaningful outcomes from this messy data. Going back to our results tab, we can look at any of these samples that way. So for example, if I take, kind of this, let's see here. Let's go to this guy. Let's go to sample number 1. And we go to its indel frequency chart, we can see, the majority of the sequences we're pulling out are still that position 0, so really no change, 64% of that still being wild type. And then we've got some -1 and then some other minor variants here, like, let's see here, this is less than 1% for this -10 position. So again, able to pull out kind of the main editing result that came from this particular guide RNA and allowing you to quickly screen and see what kind of results this guide RNA is providing for you. Now, the second experiment type that we're going to do, and I'm going to start a new project, load up, is going to be clonal screening. And so if we switch our toggle over here, the software is going to kind of take in different parameters for this analysis, but ultimately the setup is very similar. So I'm going to be loading in our control file here. So we drop in our single control file. We're going to be dropping in 95 edited samples here because we're going to be screening an entire 96-well plate of samples. And you can see how fast that was uploaded and is being validated by the software. And then I'm going to be importing a mapping template from my computer as well that's going to identify all of those particular samples. So this is our clonal mapping template. And you can see for this example, we have our guide RNA sequence, which is consistent for all of the results here, as well as a donor sequence that's been incorporated here to be used for doing a specific SNP change for this particular example. I'll go ahead and click Analyze, and it will do the analysis for all 95 of these samples. All right, and now we have our results here. We've got a very long table, a whole lot of results here that we need to look at. But what's nice is that it's going to be auto-sorting this based on this grading system that we've put into place, looking at that percent editing, looking at percent HDR, and that edit diversity. So if we take a look at kind of our sample here that was in well H1, we can see that we've got a very clean result here. So if we look at our plate view, we can see in this 96 well plate view, all of our samples that we were looking at. We've got our blue ones that are kind of maybe our less likely to be successful samples. We've got our stars here, which are going to be our ideal case scenarios. We've got our control file here. We've got a well that's a true wild type, very low editing efficiencies. And we can see here our kind of our top of the list guy here that has a model fit of 1. We've got we've got 100% editing efficiency, and we've got that diversity of edit being 1. We've got only 1 outcome from this, and so we have, we look at our indel frequency chart. We have just that SNP change occurring. We have a very clean outcome here for the editing outcome that we were looking for. If we go back to some of these other examples here, we've got a diversity of edits of 2, and I go in and look at this indel frequency chart. I can see that we have some examples here where we have our successful knock-in with that SNP change, as well as some other ones and some minor variants here where we've got that +1 insertion. So we've got 50% being having the SNP correction done, and 50% with this, with a single base insertion here. Let's see here. Let's see, what was I going to do? And then for reference here, the SNP change that we're trying to do, just to make it super clear, is at this position here, we're doing a C to G mutation with this SNP. So we've got our edit site here, and going back about 9 bases, I believe we got our SNP change here. For our knock-in examples, you can do a donor to control alignment, so you can see how well your donor lines up with your control sample. You can see that G to C base being highlighted here. Those are where our changes are, so that we know what our donor was specifically trying to do. And then we can also see our sequencing traces as well if you need to look at that raw data. Going back to our plate overview here, again, we can look at a glance at our 96-well plate and clearly see, our strongest candidates from our clonal isolation and moving them forward. This has been a great, powerful tool for a lot of our custom services team that supports cell line model generation, and they were the alpha users for this software, and has definitely improved the efficiencies of those systems. So it's really a great platform for that. Yeah, and with that, I'm going to switch back to my PowerPoint for some closing remarks and our Q&A. Just give me 1 second here. All right, hopefully you guys can see my PowerPoint again, and we'll just wrap up here. So again, kind of to wrap things up as a summary, we have our TrueDesign Genome Editor, being able to support all of the design input needs, and take those considerations for your experimental needs, identify the appropriate sequences, and provide you kind of a complete solution to have a successful edit at the bench. And then we have our Seq Screener confirmation app to be used kind of as an analysis tool after your gene editing is done for both primary screening, kind of understanding how well a guide RNA is going to be working, how well your donor is being incorporated into a pool, and then as well as supporting kind of that clonal isolation and screening workflow as well when you need to be analyzing, dozens of possible clones and figuring out which ones successfully have your specific SNP changes or specific editing outcomes. And again, in summary, we did a quick review of our genome editing and the R&D data that went behind this. We did a demonstration of TrueDesign Genome Editor, and then we wrapped up doing a demonstration of the Seq Screener Gene Edit Confirmation app. And with that, I thank everyone for their attendance and looking forward to answering your questions.
All right, that was great, Matt. Thank you. We are just about at time, but I'll go ahead and we'll fit in some of these questions that have come in. If you still have a question that you'd like to ask, you can still do so. Just click on the Ask a Question box located on the far left of your screen. I'll have Matt answer a few now, but any of those that come in that we don't have time to answer, and then anything that gets submitted during the on-demand viewing period, we will address them using the contact information you provided at the time of your registration. Okay, so Matt, there's a handful of these. I will try to pick some that are going to be most relevant to the biggest group. Okay. Here's one that I know we've heard once or twice. Why do I have to log in to use these tools?
Right, yes. So both the TrueDesign and the Seq Screener do live on our Thermo Fisher Connect platform. We do require registration access those. It is not really our intent for it for marketing purposes, but we do have the opportunity, like I showed for SeqScreener, for that data to be saved on our cloud platform so that you can access it. There's also a whole variety of other applications that I didn't talk about in this presentation. And since this is kind of our larger ecosystem of bioinformatic tools, they do require a login for that. Just a reminder, it is just a first name, last name, email, and password. You don't need to be providing, kind of all of your institutional info. If you have a purchasing agent that does stuff for you, obviously you can still go through those channels. It's really just some basic identity information to access the tool. And so we hope that is suitable for you guys to access kind of the power here.
Right. So one kind of sounds like a usability question. What is the difference between knockout with indel and a deletion? So I think this is speaking to some of the options on the TrueDesign tool. Right.
So what the deletion is what we're supporting with the deletion functionalities there is to do a very targeted removal of bases. Now, like the idea for that would be like if you just needed to remove a series of amino acids and still keep the rest of the functional protein. By the knockout by indels, you can get that outcome, but what's more likely to happen is that indel is going to be disrupting the reading frame and leading to a completely truncated protein or a bunch of missense mutations that's going to lead to the entire protein being degraded. The deletion settings there is really to try to make that targeted to do specific amino acid removals while still keeping the larger functional protein and functional sequence intact. So it's kind of more of a precision removal of DNA versus kind of a more blunt-ended response of using indels to cause knockouts.
Got it. And then the other distinction then, though, I suppose, is that the knockout with indel is simply we're making a cut and letting the cells repair themselves.
Exactly. Yeah. Exactly.
Whereas the deletion uses a donor.
It uses a donor. Yeah. And
okay.
Thank you for that. Yeah, and you're right. So the deletion where we're doing those specific amino acid removals is going to have a donor included with that to tell the cells kind of how this needs to be repaired. When you're doing this as an indel, it's in the absence of a donor. So the cell is just repairing it by that non-homologous end joining somewhat randomly, depending on the cell type.
All right, makes sense. Okay, so here's one for the second tool that you showed. Does SeqScreener only work on Sanger sequencing?
Yeah, so SeqScreener is a Sanger sequencing analysis tool, so it's specifically for doing data convolution from those kind of base calls that come out of the capillary electrophoresis. Now, it will use any .ab1 file that comes out of any genetic analyzer. It doesn't have to be a Thermo Fisher piece of equipment to do that, but it is specific for Sanger sequencing analysis of genome of gene editing events. There are other tools that are in development around the next generation sequencing platform, and we hope to be sharing those with you in some future webinars.
Excellent. All right, well, we've gone a little bit over. Hopefully some people have hung in to hear their questions answered. No, that's fine. So thanks again for your time and showing us these great tools that are available. We also need to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. And then, of course, thanking the audience for joining us today. This webcast is going to be able to be viewed on demand, so LabRoots will alert you by email when it's available for replay. So please, we encourage you to share that email with your colleagues or anyone who may have missed today's event that might be interested. And until then, have a great day. Has a hard